Well, as you see here, there are a lot of people working. We are not mathematicians. Here you have veterinarians, biologists, statisticians, engineers in water resources, chemists, and so on. So this project became quite large, starting with a very simple first idea, which was a problem that the biologist and the veterinarian brought to us, and that was to build a model for fish dynamics in these shallow lakes. They wanted to create some tools for management of the shallow lakes, and particularly for exploitation of fish. When we started looking at what they had, we realized that it was not possible to build a model for fish dynamics if we didn't know what the characteristics of the environment were. Why I say this? Because they showed us data, for example, from two very similar shallow lakes, similar in size, surface, depth, in water quality, but they had very different production of fish. One had an abundance of fish and the other didn't. So we realized that something else was behind the dynamics of the fish, and we needed to know better what was going on in that environment. Now, this started to grow, and I will show you what were the problems. This is a satellite image of the province of Buenos Aires, and you can see there are plenty of water bodies. Those are the shallow lakes. There are, in fact, many more than anyone can think, even people living there. And you have more than 10,000 larger shallow lakes, larger than 10 hectares, and 146, even more than that, between just a small size and up to 10 hectares. When we looked at past information, the literature, we could see that Ringelé, which was a very respected limnologist, had classified the shallow lakes into these many groups. And here in this area, there weren't any shallow lakes. This was a dry area, going to what people called the desert, which is the dry pampa. And quite recently, this area looks like this. And this is because of an increased rainfall in that area. So we started to see that the dynamics of the shallow lakes had something to do with precipitation patterns. Now, when we look at the characteristics of these shallow lakes, you can see that they have low depth that determines instability, accelerated biochemical cycles. They're polymictic, that is, they're well mixed. They have flat bottom and white littoral, the border. No thermal or chemical stratification. They have low transparency. They're colored green, brown, or clear. They're called like that. This is a brown shallow lake, a green shallow lake. They change colors. And that's one of the main characteristics. They have, at some periods, a lot of chlorophyll, which make them green. Sometimes they become clear, and then they turn back to green or brown, depending on environmental conditions. They're highly variable in water permanence. Some are quite stagnant, others are not. Salinity changes a lot, and this is very stressing for fish because of the changes in water level. salinity concentrates or dilutes. I mean, salt concentrates or dilutes. So this is highly variable, and the range of variability is large. So fish had to adapt to that condition. They're naturally eutrophic, very productive, that means phytoplankton, surrounded by reed, tend-to, marshes, or dry salt pockets. Now, this is something that is also related to climate. When they start shrinking in a certain way, they go through different trophic phases. And when you have a cycle of wet climate with lots of precipitation, it seems like this is in the terms of a biologist, they reset, they start again, like a newly formed water body. So from the point of view of the life forms in those shallow lakes, it's very interesting, and it has a lot of variabilities in many different aspects. They are hydrologically unstable. You have some permanent shallow lakes, others that depend on the seasons. Some are temporary, some just for a short period. There is instability of water input, changing shape and size because of the precipitations. Both water and sediments are disturbed by winds, and this is what makes them brown when you have the wind pulling up all the sediments. Chlorophyll values are really very high, very, very high compared to other shallow lakes, even in South America. Here you have a sequence of satellite images just to give you an idea of what we mean by variability. Okay, and here you have some pictures. Different dates, different years, how they can just disappear or increase their level incredibly. Okay, so we shifted objectives. We said, okay, before we can even think of a population dynamic model, we need to know more about the structure and the functioning of the natural and socioeconomic components of the shallow lakes. This is because people tend to throw residues into the shallow lakes. We need to develop new observation and monitoring tools and integrate field data and remote sensors data because of the numerosity of shallow lakes. You cannot go and sample every shallow lake. You have to find a way to monitor them remotely, in a different way. And propose management strategies for shallow lakes, taking into account socioeconomic, environmental, and cultural aspects that have an impact on their dynamics. So we focus on these two: developing observation and monitoring tools and integrating field data and remote sensing data. Okay, the study sites. We had to choose shallow lakes, and we selected them based on their hydrological characteristics, location, accessibility, trophic structure, site use, so what activities we could find around them, and basin land use. There's another image here. This is the southern part of the province of Buenos Aires, and we chose shallow lakes that are up in the basin, the river's basin, so that they are less contaminated than the ones that could be found closer to the end of the rivers. So we picked up, well, we have been monitoring many of them, but for this study, this particular study, we just picked 8 of them. We monitor several variables: physicochemical parameters, nutrients, plankton, macrophyte, fish, because the biologists are interested in those and want to correlate what's going on in the shallow lake with the other parameters and the fish. And in some particular cases, heavy metals, pesticides in water, and sediments and organisms. While for the fish, they consider diversity, percentage of captures, captures per unit effort of one particular species that is very valuable commercially, condition, growth, size distribution in captures, gut contents, average length and weight, and gonads. Now, first of all, a statistical analysis of the data was performed for these shallow lakes, particularly values of chlorophyll, A, total solids, and Secchi depth. These are the classical parameters, the 3 parameters that are usually considered for analysis of the trophic state of the shallow lake.
The Secchi disk is for optical properties?
Yes, you have a white disk with a rope, and you submerge it, and you measure how far it takes to lose sight of it. So that gives the turbidity of the water. It's a good measure. It's very primitive, but it still works fine. Here you have the depth, minimum, maximum, and mean for all of them. And you can see that these are quite variable. For chlorophyll, you can have a large variability from one measurement to the other, and total solids just the same. An initial classification was performed defining the quantile probability distribution. So they were divided into 4 classes, 4 categories defined by class 1 formed by clear water shallow lakes, class 2 by shallow lakes presenting intermediate turbidity values, class 3 mainly the green ones, high values of chlorophyll, and class 4 highest values of chlorophyll and total solids, so sediments in general. These are the values for each of the classes. Now, we wanted to use this classification to develop some tool, some algorithm that could allow us to classify all the other shallow lakes from satellite images, taking these as ground truth value. So, absorption of light means suspended clay particles. Dispersion of light means dissolved organic substances. And if you add up absorption and dispersion, you can detect phytoplankton from satellite images. Now, how we use those? If you have, I'm sorry, this is in Spanish, you have the generalized spectrum signature, spectral signatures, and the reflectance depending on the wavelength in the image. So we have the spectral signature of soil, like this, and we have the vegetation spectral signature, and we have water. This I showed you yesterday. Now, if we take Landsat images, for example, what we have are the bands here, band 1, 2, 3, 4, and 5, which are the optical, and the last one is thermal, infrared bands that allow us to detect what's in the water. Let me show you this. This is a composed image. It's a fake color image of the coast of the province of Buenos Aires, where a city, a small city. And this is what you see from band 1, band 2, 3, 4, and 5. That means that you don't get any colors, it's just intensity. Intensity of the reflectance in each one of the band frequencies. So with that, what we do, this is the process. You have the original images, you select the area of interest, you recover the image flows, you do a co-registration so you can overlap images and compare them from different dates. You have, well, I explained to you yesterday the tassel cam transform that allows us to cut off the water and see where the water is. From that, we can individualize the water bodies, and from them, we can monitor them. And on the other hand, we can use the spectral signatures with a neural network classification to pick up the classes, those 4 classes that I had mentioned before, or concentrations of chlorophyll or total solids, or both. And on the other hand, with spectral and mixing, we also try to detect the classes and see which one of those 2 methods is better. Okay, so how do we select the area of interest? For example, there we have a shallow lake. It's called La Brava. It's one of those, the one that is clearest. And we do tassel cap. This is the wetness image. You see Brava is here. We have the brightness image that gives us the soil. And then we subtract them and create the water mask. Why is it important to do this work? Well, here we can identify La Brava, and here you see it's dark, it doesn't reflect as the soil reflects. We have other water bodies here, and you can see them there. But let me show you something more. You see, those are the ones we have picked up. But here, for example, in this place, there seems to be a water body here. So if we do the subtraction, we see that it's not a water body. Okay, it's a false suspect, false candidate. So with the water mask, we can see how the shallow lake changes as time passes. So we can picture space and time.
What was the time scale?
Well, here in the time scale, it's not even because it depends a lot on how many clouds you have when the satellite goes by. So sometimes if you have clouds for a long time, it's very difficult to pick up any information.
A few weeks or a few years?
No, months.
Okay.
We usually want to have the images at the same date, but no more than 3 days away from the sampling, the monitoring, when people go and pick up the samples. So we can really find the information to be useful. If you wait more than 3 days, then it's not reliable. So with those sequences of images along the year or along any given period, you can prepare recurrence maps where you have the percentage of times that this pixel appears as water. So you have an idea of how much they last, or what the frequency or the lifespan of the shallow lakes is. Now, this is the spectral signatures of these 8 shallow lakes at different times. So the spectral signatures do change, and what we want to do here is classify them by spectral and mixing. Now, in order to do spectral and mixing, as I showed you yesterday, you need to find or to pick up or choose the bands where the signatures that you're going to use as reference are the most different, so as to have a basis for your space of Ed members. So here, what we can see is that this blue signature corresponds to La Brava, which is the clearest one, the most clear water, while the red ones correspond to La Peregrina, which is the one that is the brownest. It's class 4. So we pick up where the bands where these two signatures are the most different, where they differ the most. And it turns out that in the case of our shallow lakes, it's band 2 and band 4. Why do I say that it turns out to be that? Because normally people working in the ocean and using this type of algorithms pick up bands 1 and 3. This sort of method has been used quite a lot for soil cover and for the ocean, but not in freshwater bodies or shallow lakes at all, because it's very tricky for shallow lakes. Now, I was mentioning earlier that because we didn't know this, that usually bands 1 and 3 are used, we just explored every band. And we realized that band 2 and 4 are doing what we need to have. So we picked band 2 and 4, and we compared the classification by spectral and mixing to the classification done with the quantile distribution. And in most of the cases, they agree. We have some problems here with one statistical classification as 1 and a spectral and mixing by 2. Here there are 4 cases in which statistically it was 4, it was class 2, but spectral and mixing picked up as 1, and here. These are the only cases that were a little bit confusing. Now, the principal component analysis shows us that reconstruction in each variable of the plane is quite good. It's all above 80. So we consider that this is a reasonable method to use for the classification. We explore another method using artificial neural networks. An artificial neural network is a black box model. We have input, and it gives you a classification, an output. And it's very useful for nonlinear systems, and very useful when you have a lot of variables. They have great predictive capacity and can ignore intrinsic noise and measurements. Now, again, artificial neural networks have never been used for shallow lakes. They have been used for other water systems, but not for shallow lakes. So we explore the possibilities. The architecture of an artificial neural network model is just like this. You have a sequence of input, and you have weights, and you have a weighted sum of the inputs that goes through a nonlinear function. Let me have it there. That is called an activation function that produces the output. Artificial neural networks are trained with an iterative process that improves the definition of the weights, and then they converge after a lot of loops, many loops in many cases, to an output. A commonly used activation function is the sigmoid, the logistic sigmoid, which has a range between 1 and 0 and 1, and 0 and 1 because you want a normalized output. So what we do is we use a forward feed called the multi-layer perceptron, and we train it using a backpropagation algorithm. You give the network input data, and you say, well, this for this frequency in this band, for this intensity in this frequency, we have this class. For this other intensity in this frequency, we have this output. For the first input is given, and the calculations through the network yield an output, and this is the error is calculated, And as the errors go down, the weights are converging into the right values. So what we do is first do the automatic preprocessing with the spectral signatures, and then we choose bands. From all the bands from 1 to 5, we picked up bands 2 and 4, and through this set of hidden neutrons that have been selected and the weights have been computed, we can determine if the output is in class 1, 2, 3, or 4. This is the architecture of this neural network.
Why didn't you start with the bands 1 to 5 and 11?
Anyway, even if we picked up for spectral and mixing 2 and 4, we tried the network with all the bands. And it turned out that for the neural network, there was a coincidence with the spectral and mixing method. The best results, the smallest errors, were computed for bands 2 and 4. Now, as I said before, the network is trained with a certain number of data and then corroborated with the rest of the data. We picked up from the 36 shallow lakes, these are numbers, shallow lake 1, 2, 10, 15, and so on. We gave numbers to them. Not to the shallow lakes, I'm sorry. We gave numbers to every sampling of the shallow lakes at different dates, because there are only just 8 and we have 36 data sets. So this means that shallow lake 1 falls into class 2, shallow lake 2 is in class 4, and so on. So each one of the shallow lakes data set falls into a class. The output is 100% accurate. The conversion is very fast. It's just 17 steps, you have 0 error. So we got enthusiastic about this and started working, doing the same thing with the values of total solids and chlorophyll. Now, for taking the intensity of the signal in the given frequency, what we did was to pick up a certain number of pixels from the image and take the average. For example, take one shallow lake, you pick up randomly 800 pixels in the image and average the intensity of the signal in a given band. And that will be the information that you are taking into account and giving it to the artificial neural network. So it's just not one pixel. It's the average of 800, 900, depending on the size of the shallow lake. So here what you have is we run together as input total solid and chlorophyll and relate them to the values that were measured in the field. And this is what we get. We have measured data are black lines, and obtained from the artificial neural model are gray. Unfortunately, the grays are not very clear here in the projection, but you can see that they agree quite well with the dark lines. This is the learning error. This gives you the convergence of the iteration process. And here you have the predicting errors when you compare the output from the corroboration data. If we take chlorophyll all by itself, run all by itself, this is the output. Gray is the model, and black is the real data. And these shallow lakes are used for calibration, and these shallow lakes for corroboration. Learning and predicting errors. You see the predicting error goes down quite fast. And this is for total solids. Again, black is real data, gray is the model. And you have learning and predicting error. Not always the convergence is smooth. Now, this is just to show you some numbers. Here we take a random sample to be predicted and all the rest as learning data. Here we take all the data for La Barrancosa for predicting with all the others as training data and so on. Here you have the mean square error and the determination coefficient. The output is quite good. Why is this not so good? Chifle Paraíso. These are extreme values. These are the brown, the very brown shallow lakes. So it's difficult to predict them very accurately if you're using as training a set that is not in the same range. I don't know if that is clear enough. They are sort of extrapolated, not interpolated. So the error obviously is expected to be a little bit larger. So these are the values for total solids, and this is for chlorophyll. And this is the regression analysis, calculated and measured, measured versus calculated, with the confidence and prediction bands. Now, we are quite comfortable with these results. Both artificial neural network and spectral unmixing give us similar results, except that for the artificial neural network, you have an estimation of the error. Now, one of the problems we have is that they all fall within the eutrophic, hypereutrophic range. And you cannot still see the differences unless you look at particular variables. And we needed some way to be able to capture which were the variables that needed to be taken into account to discriminate among those shallow lakes and see what were the differences that are not really visible from this classification of the concentrations of chlorophyll and total solids or SECI-DEF. These three variables usually taken into account, chlorophyll, total solids, and SECI-DEF, are not sufficient to separate, to discriminate among the shallow lakes. So we needed to go deeper into the chemistry of the water in the shallow lakes. So we took a different set of shallow lakes. There are more shallow lakes here. We went a little bit further, close to the sea, and we took some shallow lake further north. The shallow lakes were sampled every month for 2 years. And then, because there was a very dry period without rains, we went again and sampled them every 2 months to really take advantage of this difference in the climate. So, the measured variables were calcium, magnesium, total hardness, sodium, potassium, nitrates, chlorates, ammonia, carbonates, bicarbonates, fluor, sulfates, total phosphorus in water, total phosphorus in solids, solids, total solids, dissolved solids, electrical conductivity, pH, SECI-DEF, chlorophyll, macrozooplankton density, and temperature. And also we considered the ratios relating some ions, and this turned out to be a very good thing to do. This is just an example of the TSI index, the Carlson's index that takes into account the three variables I mentioned, chlorophyll, total solids, and SECI-DEF. And for example, here you can see these have low values on almost every variable here, Secchi, total phosphorus, chlorophyll, total solids, with a small area. But here in this shallow lake, you can see that the values are extremely high. These total solids values are extremely high, as chlorophyll does. And this is this is the very brown one. These two shallow lakes were shrinking. They were losing water. They were becoming smaller. Cases that are considered upper extremes following the usual classification are in fact lower extremes when you look at Pampas water bodies. So the usual methods really don't apply. A principal component analysis was performed on the variables together with path analysis, permitting the reduction of variables just to find the correlations among them and reduce the number of variables to look at. This was followed by discriminant analysis with the purpose of distinguishing groups of shallow lakes and identifying the most important parameters that can describe them. And while this is the most important variables. And then we defined another index that we call Pampas Shallow Lake Eutrophic Index, defined as the first discriminant function standardized by common variables weighting the presence of total solids, chlorophyll, and carbonates as principal determinants of the trophic state. And this is the combination. The weights are here. And these allow us to discriminate. You see here, using Carlson's TSI, all the shallow lakes look the same. They are all in a very, very small range. This is the very light, clear one. This is the very brown one. But with the new index, the new function that we propose, you can separate them into groups. And then we try to explain what makes these groups to be formed in that way. We made a sensitivity analysis, and it showed that the proposed index is moderately sensitive to these variables. And this is the important one, the one that helps us distinguish between them. This is a particular characteristic of the Pampa shallow lakes, this ratio of magnesium versus calcium. I'm sorry, I'm not a chemist, so I cannot explain this properly, but it has to do with the process of sedimentation and how the ions are exchanged and how they regroup after wind picks up all the solids up into the water. It's a very particular process of this region, the calcium and the magnesium relationship. Naturally, the index is very sensitive to chlorophyll. It's very reasonable. And in all cases, this ratio was above 1. But for example, from one group to another, this shifted from 1 to 2. So it does make a difference. By analyzing the standard error, it is readily seen that some of the shallow lakes exhibited a higher variation in the trophic state, while others were more stable and show a regular behavior during the study period. They didn't change much. An inverse relationship between water volume and the calculated index values was observed, linking precipitations to the degree of eutrophy of Pampa shallow lake. For this, it was extremely important to have the data for the dry period. That helped us a lot to make this connection. It seems reasonable to pursue investigations on how floods and droughts affect the degree of eutrophication of the shallow lakes. Now, this is just the beginning, because now we are working with meteorologists and physicists in a project that has been recently approved. We got the grant to analyze how the behavior, the patterns over the oceans, both Atlantic and Pacific, have an effect on these dynamics. It's not correlated directly to precipitations during the year, during the time of the observations. But there is some evidence that there is a relationship when you look backwards a couple of years or more. So you need a sort of accumulation of information that makes the shallow lakes behave in a certain way. On the biological side, the index helps identifying the dominant trophic webs and their possible evolution, both in the case of environments where the plankton chain dominates or those in which the detritus determine the main energy path. So from the biological point of view, this classification also has as a meaning. And as you look at the values of our index and see how the water body evolves into higher values, it starts dominating the detritus. The detritus dominate. And as the index gets to lower values, it's the plankton chain dominating the behavior of the food chain. Now, just to put everything together, the mostly eutrophic and hypertrophic lakes challenge the usual methodologies because of the influence that these characteristics has on the optic properties captured by the satellite sensors. Spectral unmixing has been applied to land cover analysis, but it has been only recently applied to water bodies. It seems an adequate tool for classifying this type of shallow lakes. Most published research shows that artificial neural networks methods have been mostly developed for water bodies that exhibit chlorophyll concentrations below 100 milligrams, and we are well above that. The least was, I think, 140 or so, and up to 1,700. Our models have proven to be very adequate for the classification of shallow lakes following their turbidity, and we have an accurate output. This methodology can be confidently used for classifying other shallow lakes in the region. The models seem capable of dealing with the wide range of concentration with low error and good confidence values. The index appears as a better, more precise indicator of the shallow lake's trophic state. It reveals the importance of carbonate and bicarbonate ions as well as the magnesium-calcium relationship that, together with the usual key variables, determine the characterization following hydrometeorological conditions. So we're trying to put together different factors, biological, physical, chemical, in time and space. Thank you.